different type of interactions which take place into the that is the interactions also. But the ultimate effect of these interactions are beneficial for the plant growth. So among these, the beneficial impact of these microorganisms are commercialized, and that commercializations, the product which comes after the commercialization, it comes under the rhizobacteria, PGPR. हाँ एक तो क्या बोला उन लोग में ज्यादा पंखा पंखा चला उन लोग ढेर आदमी का ये उसमें हम नहीं देंगे इतना लगी नहीं दोनों मिलके बोला था हमारे 550 दिया 300 दिया बोला कि हम नहीं देंगे आप दूसरा जब ढेर आदमी का पानी ज्यादा खर्चा हुआ उन लोग हम क्या बोले Now is it visible? Hello.
Yes, sir.
Okay.
Yes, sir. This is visible.
Okay. So today we will discuss plant growth promoting rhizobacteria. So these rhizobacteria are mainly the ability of certain bacteria to colonize in rhizosphere very aggressively. Okay. Okay, so plant growth promoting rhizobacteria. So these rhizobacteria are bacteria which reside in the rhizosphere, and the property of these rhizobacteria are they grow very fast into the rhizosphere. Okay, so the rhizosphere bacteria that exert a beneficial effect on plant growth are called plant growth promoting rhizobacteria, and these bacteria have ability of certain of these bacteria to colonize in rhizosphere very aggressively. They colonize very fast into the rhizosphere. Okay, so that's why the beneficial effect of these rhizobacteria takes place into the rhizosphere, and they are taken up by the microbes, plant roots also, and even their activity are able uh, to protect plants from the different disease. Okay, so that's why they improve the plant growth, and and in this way they affect the plant growth also for the beneficial effect. Okay, so these rhizobacteria have ability to grow very fast into the rhizosphere. They grow very aggressively and colonize in each and every part of the rhizosphere or on the rhizoplane. Okay, so rhizosphere bacteria exert a beneficial effect on plant growth and are called plant growth promoting rhizobacteria. Okay, so these terms, plant growth promoting rhizobacteria for the beneficial microbes, was first at all given by Klopper and Scott. Okay, in 1981. So first time this term was coined by Klopper and Scott in 1981. If you see the beneficial effect of these microorganisms as PGPR, the Pseudomonas bacteria are most widely used for the plant growth promoting rhizobacteria. Different species of Pseudomonas are available. They are abundantly found into the soil also, rhizosphere also, and they have several beneficial effects on the rhizosphere. Okay, so there are diverse PGPR strains have been used successfully for crop inoculations, and during seed treatment, or in the when they are applied into the rhizosphere, their beneficial effect are commercialized also. Okay, so if you see the example, so all the nitrogen fixing microorganisms comes under the rhizobacteria, PGPR. Even the phosphate solubilizing bacteria and other beneficial bacteria, potassium solubilizing mobilizing bacteria, even Azospirillum, Bacillus, Pseudomonas, Rhizobium, Serratia, Astenotrophomonas, and Streptomyces. So all these microorganisms PGPR, and different strains of these microorganisms are mainly utilized for the PGPR. So these microbes, these strains of the microbes are isolated. They multiplied into the laboratory, and after laboratory, after multiplication, they are formulated into a based materials or liquid based materials, and then they are marketed into the market for their use into the farmers field. Okay, so there are some fungi also that come into the genera Inflomyces, Corneotherium, Trichoderma. They have also some beneficial effect. Also, Aspergillus is also comes under this category. So, bacteria, fungus, and actinomycetes, all these group of microorganisms are mainly utilized for the plant growth promoting rhizos.
Excuse me, sir.
हाँ बोलो.
सर एक स्टूडेंट को ज्वाइन करना है, कर दीजिए.
Okay, so the most important bacterial genus which are mainly exploited into the PGPR are Pseudomonas and Bacillus. Okay, so Pseudomonas and Bacillus genera are the most commonly investigated and exploited PGPR, and often the dominating bacterial group in the rhizosphere. If you see the abundance of microorganisms into the rhizosphere, then Pseudomonas and Bacillus are dominating into the rhizosphere, into the rhizospheric soil. So these microorganisms are also have.
Excuse me, sir.
Okay, so we are discussing is visible.
Yes, sir.
Yes, sir.
Yes, sir. So we are discussing about the most dominating bacteria which are found into the soil, that is Pseudomonas and Bacillus. They are abundant into the soil, and they have also different type of activity, different type of biochemical properties, through which they are most commonly investigated as the PGPR, and they are exploited also for the PGPR property, and are often found very much high population in the rhizosphere. So Pseudomonas fluorescens and Pseudomonas putida, they are the two microorganisms which are when applied on the potato tubers and sugar beet and radish seed treatments, where they have beneficial effect on growth and yield of the plants also. So these Pseudomonas and Pseudomonas fluorescens and Pseudomonas putida are comes under the PGPR, plant growth promoting rhizobacteria. So when they are applied either seed treatment or on the tuber treatment, they have beneficial effect on plant growth, and you can achieve the higher yield of the crop when they are used. These PGPRs are used. So one of the important PGPR which are marketed by the USA, that is Quantum 4000. That is the first commercial product of the PGPR, that is marketed by Gustafson Incorporations, Texas, okay, from USA. So these PGPR Quantum 4000 is a growth promoter. They promote the growth of the peanut and cotton, okay, and this product Quantum 4000 contain mainly Bacillus subtilis. The strain of Bacillus subtilis is GBO3. Okay, so these DBO3 are capable of producing endospores and tolerating heat. Heat uh, can suppress soil-borne disease also. So they are able to control certain soil-borne disease, and through which the growth of the peanuts and cottons were improved. The yield of the peanuts and cotton were improved by the use of simply the treatment with the Bacillus subtilis, strain DBO3. Okay, so this product was commercialized as the first PGPR product in 4000, and it is marketed by USA, that is Gustafson Incorporations Limited, Texas. Okay, the another one is the fluorescent Pseudomonas. Okay, they are able to control tackle disease of wheat and fusarium wilt of radish also. So they can also be used as PGPR. The mechanism through which fluorescent Pseudomonas control the tackle disease of wheat and the wheat, they mainly mop up the nutrient in the rhizosphere. They mop up the nutrient in the rhizosphere, mainly iron. Okay, so through the mop up of nutrients in the rhizosphere through the cytochrome production. So when cytochrome production takes place, they remove the iron from the soil, or the availability of the iron for the pathogenic bacteria or deleterious type of microorganisms are restricted or limited, and that's why the growth of the deleterious type of microorganisms are affected. Okay, so the point of emergence of lateral roots are favorite spots for the DRBS, mainly deleterious type of bacteria, fungi, or microorganisms, and PGPR appear to compete for these spots very effectively. Okay, so that's why they are able to control the take-all disease of the wheat in case of fluorescence Pseudomonas. Okay, so many bacteria have these potentials. 1 minute, please. So many bacteria have this type of potential, and they have ability to control the disease as a biocontrol agent, and operating by the secreting antibiotics and by other mechanisms. There are different mechanisms, in which one mechanism is the secretion of antibiotics. So when the secretion of antibiotics takes place in very small quantity, they are able to affect the growth of the surrounding microorganisms in the rhizosphere, and that's why they are mainly used as a biocontrol agents. Okay, like echinoplenes, agrobacterium, arthrobacter, bacillus, azotobacter, Enterobacter, Rhizobium, Bradyrhizobium. So all these type of microorganisms are there, and these are able to control the pathogenic microorganisms by the different mechanisms. One mechanism is the secretion of antibiotics also, and some mechanisms are also there by development of cytophores, even production of SCN is also there. So all these mechanisms are helpful in controlling the in controlling the pathogenic effect of the microorganisms in the rhizosphere. Another example is Agrobacterium radiobacterium bacter. Agrobacterium radiobacter, that is a strain 84. So this bacteria is mainly used for the control of crown gall disease caused by agrobacterial tumefacients through agrocin 84 antibiotics. In 1980 1990 these type of product are very much commonly available into the market as the agrocin 84 antibiotic powder. So these antibiotics, when applied into the in the rabi crop, they are able to control the crown gall disease also in case of wheat also. So agrocin 84 antibiotics are mainly used. So these agrocin 84 antibiotics are mainly extracted from Agrobacterium tumefaciens. Okay, and the 84 strain was 84. Agrobacterium radiobacter strain 84. That is the excellent example of a biocontrol agent controlling crown gall disease caused by Agrobacterium tumefaciens through agrocin 84 antibiotics. So this is one of the commercialized products which are very much marketed into the in our country also, and very much in use in the 90s. Okay, the mechanism through which they are able to control our SCN production. So hydrogen cyanide produced by the many microorganisms, okay. So Pseudomonas application as PGPR increase potato yield by reducing SCN production by DRMOs through the cytophore mediated competition for the iron which is required for the SCN production. Okay, so this is one of the mechanism through which the potato yield was improved if they are using the PGPR mitigated mechanisms. Okay, so SCN produced by the many DRMOs reduce potato yield. But when Pseudomonas applied or Pseudomonas treatment, seed treatment or root treatment has been done as a PGPR, they increase potato yield directly by reducing the SCN production by the DRMOs. The DRMOs production reduced as all the rhizoplanes or rhizospheres are colonized by Pseudomonas, and they restrict the growth of the DRMO, and then due to the reduction in the growth of the DRMO, SCN production reduced, and so the potato yield was enhanced. Okay, so it is one of the mechanisms through which iron production was restricted through the cytophore mitigated mechanisms. Okay, so this is one of the example. If you see cytophores, that is cytophores also called iron sequester. Right? So iron sequester means they mop up the iron, or their bioavailability was reduced for the pathogenic microorganisms. Okay, so cytophores, which are low molecular weight compounds, and they have high affinity of iron, they act as a chelator and that transport iron into bacterial cells, and are produced by many of the bacteria which are also known as the PGPR. Right? So the most biocontrolled agents are working on this principle also. Okay, so cytophores, which is low molecular weight compounds, have high iron affinity chelators that transport iron into bacterial cell, are produced by some of the microorganisms to solubilize and competitively acquire iron and iron limiting conditions created, which making iron unavailable to other soil-bound pathogenic microorganisms which cannot grow for the lack of it. Okay, so through the cytophore production, the bioavailability of iron is reduced, but this bioavailability of iron is reduced for the deleterious type of microorganisms or for the pathogenic type of microorganisms. Okay. The another example is fluorescent pseudomonars that produce yellow-green fluorescent cytophores. Okay, so these fluorescent pseudomonars produce yellow-green cytophores, fluorescent cytophores. Then PGPR function better in neutral and alkaline soils than in acid soil. Okay, so because PGPR mainly bacteria and they work better into the alkaline conditions. That's why they are work better into the neutral and alkaline conditions. Why the name is fluorescent pseudomonas is given? Have you any idea? Is it audible?
Yes, sir.
So, yes.
Maybe due to the bioluminescence.
Yes, yes. Some illuminations takes place, and that can be observed in the uh, uh, under certain conditions. They can be observed in the laboratory conditions, and through which they are identified also. Okay, so that's why they are fluorescent pseudomonas, and these have a specific property for the certain a specific group of microorganisms to identify them. Okay, so I have told you that PGPR function better in neutral and alkaline soil, as neutral and alkaline soils are better condition for the bacteria. Okay, than the acid soil, which is better condition for fungus. Okay. Now, agrocin 84 antibiotics that is produced by the Agrobacterium radiobacter, which is controlling the crown gall symptoms also. Another one is the antibiotics, which are several type of antibiotics are produced. The examples are phenazine type of antibiotics, which are produced by the Pseudomonas fluorescens in the control of take-all disease of wheat also. Okay, so fluorescent Pseudomonas, that is a specific wheat of Irvinia carotovora, that causes soft rot disease of potato. Okay, so in the market, if you see, several type of bioproducts are available into the market as a PGPR, and they can be used as a seed treatment or a root treatment. Even for the foliar spray, these are also available for controlling the disease or for the better yield, for boosting the yield of the different crops. So they are also crop specific also. Several antibiotics are produced by the microorganisms in the rhizosphere through the PGPR, plant growth promoting rhizobacteria, and they have different types also, like heterogeneous group of organic low molecular compounds. These antibiotics are there. Phenazines, that is deleterious to the growth and metabolic activity of microbes mainly. Some other examples are fluoroglucinols, polyuterones, pyrolintrins, and cyclic lipopeptides. So these are antibiotics, mainly bacterial antibiotics, which are mainly secreted in very small quantity in the rhizosphere, and due to their pergency to the rhizosphere, they are able to control certain plant pathogens or soil-borne disease also. Bacteriocin, one of the narrow spectrum molecules, and these narrow spectrum molecules are new microbial defense system also, and effective for very similar type of microbes. So for specific disease controls, for the control of a specific microorganisms, a specific plant pathogens, or a specific soil-borne disease, these bacteriocins are mainly used for the control of these disease, and through the control of these disease, we can take a good crop after the application of these antibiotics. So that's all about the PGPR, plant growth promoting rhizobacteria. Another important concept in the rhizosphere and phyllosphere is the phyllosphere and phylloplane. Okay, so as we have seen that in the rhizosphere, microorganisms are able to colonize into the rhizosphere in the root zone due to the presence of certain growth promoting substances, like the food materials which are present into the rhizosphere into the root zone. these are in the phyllosphere. In the same way, the microbial growth takes place, although their populations are very less that depend upon the food requirements or food present for their growth. So very a small amount of colonizations are there, and certain specific places in the phyllosphere and phylloplanes are there. Okay, so if you see, bacteria is present or microorganisms are present everywhere. I have told you it is present on the leaves also, on the plant surface also, on the books also, on the hands. Everywhere these microorganisms are there, but their colonizations will takes place only when their food materials are available there. So on the plant leaves also, on the plant surfaces, stem surfaces, where certain specific spots or some cuticles or some type of spots are developed, and if certain food materials are there present there, then colonization of these microorganisms will be also takes place. Okay. So these are mainly due to the establishment of typical flora on leaf surfaces added by the cuticle, waxage, and appendages which help in the anchorage of microorganisms. Mainly in the cuticles, the waxage and appendages which are developed, they are the places for the anchorage of the microorganisms. Their extent of influence depend on the material of leaf diffusates or exudates. Like in the rhizosphere, the root exudates or root secretions or the organic matter contained into the rhizosphere or root pruning or dead roots or cell lysis are important for the rhizospheric microorganisms for their growth and development. In the same way, for the rhizospheric phyllosphere microorganisms, extent of influence depend on the material in the leaf diffusates or exudates which are secreted in the phyllosphere or phylloplanes. And the principal liquidative factors in these exudates are mainly amino acids, glucose, fructose, and sucrose. Okay, so these materials are work as a food material for the microorganisms in the uh, phyllosphere. So if the catchment area on leaves or leaf sheaths are substantial, then these habitats provide nichage for nitrogen fixation and secretion of substances capable of promoting the growth of the plants also. Okay, so if these nutritive factors are present on the exudates or on the leaf diffusates or several waxage and appendages cuticles are there. So if the catchment area on leaves or leaf sheaths are substantial, then these habitats provide niches for the nitrogen fixation and secretion of substances capable of promoting the growth of the plants also. Okay. So the leaf surfaces has been termed as phylloplanes, and the influence of the area the zone on leaves inhibited by the microorganisms as comes under the phyllosphere. These terms phyllosphere was coined by the Dutch microbiologist Reunin. So she has mainly coined this term phyllosphere when she was working in the forest for the microbial populations study in the rhizosphere and phyllosphere. So the term was given from her observations on Indonesia forest vegetation, where thick microbial epiphytic associations were found on the leaves also. So first at all, they observed that the leaf surfaces and shoots are also influenced by the microbial populations, and good microbial populations are available on the vegetations also, and thick microbial epiphytic associations were there. So that time they given the term phyllosphere. Reunin, she had given the term Reunin, phyllosphere. Different nitrogen-fixing bacteria such as Begerichia and Azotobacters were found on the leaves also. Besides Pseudomonas, Pseudobacterium, Phytomonas, Irvinia, Sarcina, and other unidentified ones have been encountered on plant surface, especially on the phylloplane. Okay, so a large group of microorganisms are found, but their populations are always less than the rhizosphere, and that will be depend upon the conditions which are prevailed on the phyllosphere or phylloplane, mainly the nutritive factor, which type of nutrients are present, how much amount of nutrients are present there, because in the absence of nutrient, they cannot grow or survive into the phylloplanes. So proper environments are required for their growth and survival in the phyllosphere and phylloplanes. Some blue-green algae like nitrogen-fixing blue-green algae, Anavena, Calothrix, Nostoc, and Telepothrix, which are encountered on the plant surfaces in the moss forest. If you go to the Uttarakhand in the several regions like Simla of the Himachal Pradesh area, several regions, if you can observe the blue-green algal growth on the plant surfaces, also on the leaf surfaces, on the stems. These growths are takes place. So they have a closed association with the plant system also, and through the nitrogen fixation, they can benefit the plants also by the supplying the nitrogen also to the plants, and other growth promoting substances are secreted, and they are able to taken up by the plants also. So several type of fungi, actinomycetes, and bacteria is there. So these are Cladosporium, Alternaria, Sarcospora, Helminthosporium, Irisifi, Podospora, Bulera, Torula, Oidium, Toxemia, Candida, Penicillium, Fusarium, Periconia, Supermospora, Mucor, Cunningmilla, Fusarium, Aspergillus, Rhizopus, Trichoderma, Heterosporium, Actinomyces, Streptomyces. So there are large group of microorganisms are mainly present in the phyllosphere, and phylloplants are there. So if you see their presence into the rhizosphere, the presence of spores of a pathogens. So not only the beneficial microorganisms, also there may be growth of the pathogens takes place on the rhizosphere, on the phyllosphere also. Okay, so when some phyllosphere, some spores of pathogens are present on the phyllosphere or on the surface of the leaves and pods, that results in the formation of substances that is referred to the phytoallergens. Phytoallergens means if some spores of pathogens are found on the plant surfaces, plants are able to secrete certain type of phytoallergens that are capable to that are bulk of the pathogens. Okay, so the presence of spores of pathogens on the surface of leaves and pods that results in the formation of substances that referred as phytoallergens. So if their concentration rises rapidly in response of the microbial infection, if microbial infections, if pathogenic microbial spores are increasing, the concentration of phytoallergens are also increased as a result of the defense mechanisms. Some fungal spores produce a chemical substances that are active in inducing the production of the phytoallergens. So there are separation of certain substances also that induce the production of phytoallergens by the host as a defense reaction to mitigate control the pathogenic. Okay, so their concentration may rise markedly in response to microbial infections or microbial infection or pathogenic microbes. So fungal spores produce a chemical substances or substances which are active in inducing the production of the phytoallergens, which including the pathogenic effect also by the host as a defense reaction. Okay, some fungus compounds are secreted by leaves also, such as malic acids in the leaves of Saccharomyces compounds in the apple leaves, some waxy materials also from the leaves of the cocoa. These materials are medicinal as a defense mechanisms for the control of the pathogens on the phyllosphere. The name elicitors has been given commonly. Elicitors that has been commonly used to denote the compounds which induce the synthesis of phytoallergens. Okay, so elicitors mainly compounds which reduce the synthesis of phytoallergens. There are maybe bacterial elicitors and abiotic elicitors also. They are mainly used. There are abiotic elicitors which include fungal and plant cell walls like lipids, microenzymes, and polypeptides are mainly used. So, abiotic elicitors like polysaccharides from fungal and plant cell walls, lipids, microbial enzymes, polypeptides, these are mainly used as biotic elicitors, which include synthesis of phytoallergens. The other one are abiotic, which are heavy metals, salt of detergent, or carbonic village also. Cold lights and UV lights are also there. Okay, so these materials, abiotics and other materials are mainly used just for inducing the synthesis of phytoallergens. Then epiphytic microbes are known. These epiphytic microbes are mainly known to synthesize all acetic acid, and these are more interesting in biological nitrogen fixation by several group of microbes. They are able to synthesize those promoting substances or also acetic acid. Some phytoallergens in different crops, they have different names. Like for the potato, sweet potato, it is caffeic acids. For the pea, it is fixating. In green bean, it is flagyolin. In the carrot, it is chlorogenic acids and 6-methoxy melanins. Then soybean, it is hydroxyflagyolin. In case of cotton, it is acifol. In the case of alfalfa, it is acifol or medicacol. Okay. There are several others. Phytoallergens are also there, which are for the different crops, and they have different effect in the control of disease. So that's all about the plant promoting bacteria and phytosphere and phyllogenetics. So that's all about the first part. क्या चीज कोरम सेंसिंग बताया कोरम सेंसिंग बताओ
सर कोरम सेंसिंग सर एक कम्युनिकेशन है सर जो बैक्टीरिया
कम्युनिकेशन एक चीज और करता है जो माइक्रोबियल पॉपुलेशन का रिस्पांस करते हैं वो डीनेटिवेशन भी करता है और उसी से प्रोटेक्शन भी होता है। डीनेटिवेशन की बात जो की है राइजोस्फीयर में उसके अनुसार से सेक्रेशंस भी होते हैं। बहुत सारे अल्कलॉइड्स जो भी डिफेंस मैकेनिज्म के लिए जो भी सुटेबल केमिकल्स होते हैं दैट आर सिंथेसाइज्ड इन द राइजोस्फीयर।
तो ये जीन जो एक्टिवेट होता है सर तो प्रॉपर पॉपुलेशन जब हो जाता है बैक्टीरिया का उसके
प्रॉपर पॉपुलेशन में होने के बाद ही वो जीन एक्टिवेट होगा, ऐसे नहीं होगा।
ओके सर
और कोई क्वेरी? चलो आज दूसरा चैप्टर एक शुरू करते हैं, बायोजियोकेमिकल साइकिल। So today we are starting a new chapter, that is biogeochemical cycle. It is biochemical and geochemical cycle, biological and chemical. So this is takes place in soil. This biogeochemical cycle, the nutrient availability into soil are maintained in soil, and we know also that the growth of the plant, the cycle of the plant system also. germinate into the soil, their growth will take place. They will flower, pods are coming and seeds are coming, and after that again, after death, their decompositions will be taking place, and after decomposition, again, the nutrients are freed from the complex compounds, and these are available in the plant system also. So these cycles are very much to the soil, and these are important for the controlling the nutrient turnover into the soil system also, and even in the ecosystem also. Okay. So if you see, there are only two systems which are going to the ecosystem. First one is the synthesis, and this synthesis it takes place in the presence of sunlight in the leaves, chlorophylls, where the sunlight converted the carbon into the different carbon molecules, 6 carbon compound molecules into the sugar compounds, and the minerals which are taken from the soils are mixed with the in the chemical reactions, and different type of compounds are formulated into the plant system, and they are transported into the plant's different parts, and the cells, tissues, and organs are developed, and the growth of the plant takes place. And ultimately, there, after completion of life cycle, there again decompositions will be takes place. So after synthesis, where the compounds are synthesized, whereas in decompositions, these compounds are again converted into simpler materials, simple compounds, and elements are again released. So only two type of processes are there, that is synthesis and decompositions. Okay. So if you see the basic requirement for the building organic massage into the plant systems, that is the carbon, hydrogen, and oxygen, and nitrogen, phosphorus, and sulfur. So carbon, hydrogen, and oxygen mainly when the photosynthesis process takes place, and nitrogen, phosphorus, and sulfur, they are taken up by the plant systems from the root systems, and they are mixed into the plant systems for the different stages. Their formations are takes place. So in soil, organic and inorganic forms of nutrients are there. They are in different forms. Some are usable form, and some are not usable forms. Plants did not take all type of nutrients, materials, or compounds, or chemical compounds which are present. Only the specific type of compounds of carbon, nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, iron, manganese, copper, chlorine, boron, molybdenum. So they have a specific chemical compounds, specific ions, which are only taken up by the plants. So some of them are usable form, and some of are not usable. So there are finite quantity of each element in the system. They are limiting into the system. They are not abundant into the system. Therefore, some system are exist to transform various nutrients into the appropriate life available forms. And these systems are mainly the decomposition through decompositions and biochemical processes. These systems are available into the soil ecosystems for the plants also. So after death of the microbes or living body or green materials, they are decomposed by microbes, and after decompositions, mineralizations of these materials are mainly takes place, and they are converted into the simpler usable forms mainly. So all elements of the microbial cells are recycled between organic and inorganic waste. They are converted into water soluble to insoluble form, or insoluble to water soluble form, or they are converted into gaseous liquids to the solid forms, or solid form to the gaseous liquids. So in the ecosystem, all these conversions are takes place. Like they may be converted from inorganic to organic, or organic to inorganic. They may be converted from insoluble to water soluble, or from water soluble to insoluble. There may be conversion of gaseous liquid to solid forms, or solid form to gaseous liquid. So this type of conversion in the ecosystems is very much important, and that are mitigated by microorganisms also through the decomposition process. So the rate of conversion from life unavailable to life available states, that are mainly commonly determines the biological activity into the rhizosphere on the soil ecosystem. So the rate of conversion from life unavailable form to life available states are commonly determines the biological activity in the rhizosphere, or the biological activity are more if the life available states of the materials are available. Okay, so the life supporting, nutrient generating, ecosystem modifying process that constitute the various biogeochemical cycles like carbon cycle, nitrogen cycle, phosphorus cycle, and sulfur cycle. So these cycles are important for the support of the life on the Earth surface. So the life supporting, nutrient generating, ecosystem modifying process constitutes the various biogeochemical cycles. Okay. Now, what is biogeochemical cycle? The process that involves with the cycling of a chemical element through its various biological, chemical, and geological forms in air, water, and soil system, and the reactions are biologically and chemically catalyzed. Okay, so these biochemical cycles are the process that involve with the cycling of a chemical element through its various biological, chemical, and geological forms in air, in water, in soil system, and the reactions are biologically and chemically catalyzed. Biologically, that means catalyzed means several microbes are able to enhance the microbial process through the synthesis of the enzymes also. So these biogeochemical cycles, act as a tool for evaluating and testing our understanding of the nutrient movement and distribution in the ecosystem. So these biogeochemical cycles it is a tool for evaluating and testing our understanding of nutrient movement and distribution in the ecosystem. Okay, so a generalized carbon cycle, if you see the carbon cycle, so this one is the one of the generalized carbon cycle conceptual model with the biological and atmospheric aspects. So as we know, the carbon dioxide content in the atmosphere is very less as compared to the soil where it is very high. Okay, and these carbon dioxide are synthesized by the different decomposition process, root expansion process, also several processes which are there. So when crops are harvested, okay, so these plant biomass, if you go in the center, the plant biomass is there. So they may be through the photosynthesis, their synthesis will be takes place, and after decompositions, CO2 evolved that comes into the atmosphere also. Even through the different process of composting and compost preparation methods or sludging methods, they may be incorporated in the soil system also. Okay, in the soil, if it is left over, they are after decomposition they are converted into the microbial biomass or humus. Okay, and through the loss of humus into the soil through the biological oxidation process, the CO2 again evolved come to the atmosphere. Okay, these plant biomass which are in the center of these generalized conceptual model, they are act as a food material for the animals also. Okay, as a feed. Okay, so plant biomass, it is taken up by the animals. They also yield certain excretions are also there, like cow dung and urines are also there, which contain the carbon dioxide. They are evolved into the atmosphere, and through the soil, if they are added into the soil, they are also comes as a part of the humus materials or microbial biomass, and again the same process will be takes place. So in the carbon cycle conceptual models, if you see, there is cycling of carbon dioxide which takes place from the plant biomass to the animal biomass, and from animal biomass to soil biomass or humus or microbial biomass to the atmosphere. So one cycling is there through which we are able to maintain the organic carbon content into the soils and microbial population into the soil for the biogeochemical functioning into the rhizosphere also. So now time is over. Okay, we will see the another model of the these biogeochemical cycle of a carbon cycle in the next class. Okay.